Good evening, everyone, and welcome to the final session of the International Technical Webinar on Advancements in Biotechnology, hosted by the Department of Biotechnology from St. Joseph's College of Engineering. We will now begin the session with a Tamil Thai Valthu. Now, I request our organizer, Ms. Yuvarani, will give the welcome address.
Hello and good evening, everyone. Welcome to today's webinar hosted by the Department of Biotechnology, St. Joseph's College of Engineering. It is indeed a great pleasure for me to welcome the speaker of today's session and all the participants, including faculties, research scholars, and students from various institutions across the country and from different nations. I also extend my warm welcome to the head of our department, my dear colleagues and students, to the last session of Day 3 of International Technical Webinar on Advancements in Biotechnology. Once again, I'm glad to welcome the speaker of today's session, Dr. Sanghamithra Saha. I welcome you, ma'am. Now, I request Ms. Swatika to introduce the speaker.
Thank you, ma'am. It gives me immense pleasure to introduce today's guest speaker, Dr. Sanghamithra Saha. Dr. Sanghamithra Saha is a lecturer at the Department of Natural Science in University of Houston at downtown Texas. She was an associate professor at the City College of Chicago before moving to Houston. She teaches freshmen to upper level courses, both face to face and online classes, such as general biology, microbial biotechnology, and bioinformatics. Dr. Saha obtained her PhD in molecular biology and biotechnology from the Indian Agricultural Research Institute in New Delhi. She did her postdoctoral research at the University of Virginia and at the University of Illinois at Chicago. She spent a sabbatical at the Temasek Life Science Laboratory in Singapore working on rice genomics. Dr. Saha's research interests are in plant microbe interaction and genomes and has been mentoring undergraduate research students in these areas. She will be talking about isolation and genome annotation of mycobacteriophages today. Now, I welcome Dr. Sanghamithra Saha to take over the session.
Hello, a very good evening to you all. Thank you, Yuvarani, for a very nice introduction. Today, I'm going to be talking about mycobacteriophages, about their discovery and genome annotation. So this is the research that has been going on at the University of Houston for the past 4 or 5 years. This is carried out our undergraduate students, mostly freshmen. So I'm going to present their work. And before we'll talk a little briefly about why we are interested in phages, what are phages. So I'm going to start sharing my screen, and if you have any questions, I'll be happy to answer them at the end of the session. So are in a university in Houston downtown, and a new building, which you can't see. The time this picture was taken was the building wasn't constructed. So a little bit about phages and why we are working on phages. So when I joined the University of Houston downtown, I had not anticipated working on phages, but I found out that the department was meeting with Howard Hughes Medical Institute and the University of Pittsburgh in isolation of phages and mutation. And this partnership is referred to as the C-Phages, which stands for the Science Education Alliance, Phage Hunters, Advancement in Genomics and Evolutionary Science. And what it is it's a 2-semester course, which is offered to, and in the process, they isolate phages, get to name them, and in many cases, they're able to submit their annotations bank and even submit a paper about their discovery. So I know most of you already know about phages, and so I'm not going to spend a lot of time talking about phage biology as such. But so by bacteriophages or bacterial viruses are very simple non-biological entities which have a capsid head and a tail. And the capsid head mostly contains the RNA or the DNA, which can be single-stranded. And as if you all know that phages can undergo lytic or lysogenic cycles. Phages we discover are one of the two. So this is just a depiction of the life cycle of phages in general, which is either they go on to infect the host cell, finally leading to the lysis of the host cell, or they may infect the host genome and thus carry out what is known as the lysogenic cycle. So, so many different, organisms in place, and so why is the interest in phages? And lately, in the past few years, this interest has increased. So phages provide a very simple system to study genes and gene expression. And as of you all probably know, they also are used as vectors to introduce genes or to make libraries. The other interest which this group is looking into is something known as phage therapy. So the what has happened in the recent years is that there are a lot of drug-resistant bacterial species which can be developed for a person who has an infection that is that cannot be treated with antibiotics. So, and what has happened also over the past few years is that there has been a decline in the number of antibiotics that are being. So, people are alternate therapies to treat these antibiotic-resistant pathogens, and so there's these 6 pathogens that are more than others, and we go by the name of escape. But we're not talking too much about the bacterial pathogens, but more about the phages. So phage therapy is an alternate promising therapy in cases where people are unable to be treated with standard antibiotics. So as the name suggests, it's a therapy in which phages are used to cure infections. And so this has been going on for a long time. This is nothing, but lately it has again revived interest in people. So in 1919, Felix de Herel, he treated 4 cases of bacterial dysentery with phages. And then in 1931, Felix de Herel also used phage therapy for the treatment of cholera in Punjab, and he got mixed results. So this use of phage therapy has been going on in Russia from the 1930s, 40s, but in the Western world, this phage therapy kind of declined. And one of the reasons ascribed to the decline in phage therapy was the antibiotic discovery and use over the past few years. So it's just that it's not popular as other treatments in the rest of in the Western world. So I'm involved in this project with the C-Phages group, and the C-Phages, what it does is it basically archives all the phages that are isolated, all the groups under the project. And so this particular case, I just wanted to focus on this because there was a girl and 14-year-old, I believe, or 12-year-old at that time. And she was a cystic fibrosis patient who had a double lung transplant. And she was fighting a bacterial infection of Mycobacterium abscessus. And no amount of antibiotics was treating her. So then by some collaboration, phages were engineered. It was a cocktail of phages that was taken from the University of Pittsburgh here and given to this girl by IV. And she was for 6 months to a year. And currently she doesn't show any infection of this area. And she's doing her life on as normally as possible. The other thing is why there are so many different viruses out there. Why work on mycobacteriophages? So the reason is simple. So mycobacteriophage, the host that we use for our experiments, Mycobacterium smegmatis, is a fast-growing culture, and it's non-pathogenic. Whereas the other strains of Mycobacterium, which are Mycobacterium tuberculosis and Mycobacterium leprae or Mycobacterium abscessus, these are slow-growing. So if you want to study them, Mycobacterium smegmatis is a good host. Also, the other thing is that if you are trying to work with these pathogenic strains, then phage typing, which is a technique will find out phages can infect this bacteria, is much faster and can be used for distinguishing between clinical isolates. So hence our interest in working on bacteriophages, and the host that we use is called Mycobacterium smegmatis or MSMEG. Okay, so I'm going to spend a little bit time talking about the research that has been going. So remember, this is an undergraduate research program, so many together work on this project. So, it's a 2-semester course. So semester, the students phages, and in the second semester they annotate the genomes. So the discovery process is simple. Students will get environmental samples and isolate phages from them. And it's easier said than done. Sometimes to isolate a phage take 4 to 6 weeks to get any isolate. Sometimes students are not successful, but many a times, I'll say 60 to 70% time, if students are persistent, they are able to isolate these phages. So this here is a simplified of the bacteriophage isolation and microscopy, which is the part 1 of the project. So you can see here on the left-hand corner, there's the isolation protocol is basically students going out and collecting soil samples or water samples and doing the plaque assays or spot tests to find out if there are phages or no phages. And that's done by looking at formation of a plaque on a media plate. So once a student obtains a plaque, then what's done is this plaque is amplified, or the phage is amplified. And if the student doesn't get it, then you have to repeat it. As I said, sometimes you may be lucky and obtain the plaque in the first go around, but sometimes you may be struggling with it for the half a semester or so. So once the plaque is the phage is obtained, then the phage is amplified, what are known as webbed plates. Webbed plates are basically plates that have so many phage particles in the plate that you only see like remnants of the bacterial cells. And so from these webbed plates, lysate is collected, which is going to be of the phage particles. And so we prefer a very high titer lysate, like of a 10 to 9 PFU, plaque forming units per ml. Reason being so that we can have enough starting material isolating the phages, phage DNA. The other thing that goes on also microscopy, electron so are sent on microscopy to see how they appear, what the preparation is, whether it's pure or not. And also the isolation of DNA that takes place. We do some restriction enzyme digest just to make initial call as to how this DNA looks like as compared to some other known samples. So just briefly talk about these steps, not going too much into of each of these steps, but the next slides are basically about these processes or steps. So the first stage of this discovery process is preparation, which students don't do, but we do. And that is to prepare the host cells and to prepare all media and buffers, etc. So again, just to let you know, so these are freshman students. So we may have about, say, 20 to 28 students in a classroom. So we have to make enough preparation so that everybody gets a chance to isolate a phage. So the preparation is quite a crucial part of this project. And allow it to grow and then check for presence of phages by infecting it with infecting host cells with that sample. So I'm not going again to too much details about how that process is done. That's pretty straightforward. So when somebody is successful in obtaining a plaque, this is what you see. You see these cleared zones on the media plate. And so these are, I would call them medium to large size plaques. You can see that they're clear plaques, and mostly this indicates that these are lytic phages. So from here onwards, what we do is, or students do, is take an isolated plaque and amplify that. So get a single population of phages. And from doing that, then we end up getting our lysate, which is high titer lysate. Then we calculate its PFU per ml and then go on to isolation of DNA from that sample. So once the high titer lysate is obtained, the next process is isolation of DNA. And the isolation of DNA is done following standard protocols. The DNA is checked for its purity, its concentration by running it on an agarose gel. And we also sometimes may do some PCR if we have primers for some clusters or do some restriction tests. So this is a slide showing how the phage DNA looks like. So the phage DNA all depends on the size of the phage DNA can vary from 50 kb to 150 kb. It all depends on the where it belongs to. So this is just a crude preparation of DNA showing how it appears on the gel, and you can see that seems to be of reasonably good quality. It's not completely sheared. And so this is just a rough estimate before we send out our samples for sequencing. So the samples are sent for sequencing at the University of Pittsburgh, who's also the partner in this Phages project. And so per class, we got 2 samples for sequencing, and they sequence the entire genome. And we can get back the sequence sometimes within a week, sometimes depends on their workload. So that process is not very long. So around time as the samples are sent for sequence, also send out samples for electron microscopes, electron micrography. And you can see there are these 3 electron micrographs of some phages that our students have isolated. So mycobacteriophages belong to this group called cordovirales, and within the cordovirales, they belong specifically to myoviridae and the syphoviridae. So myoviridae phages typically have this isometric head, which is seen over here in the big swole. So this is the isometric head here. And this is the tail. So you can see compared to the other 2 phages, these tails are much longer in these 2 phages as compared to the syphoviridae. So also another thing to be, which we didn't know until we got our sequence sample, until we got our phages sequenced, was the size of the DNA within this capsid. So of big swole, when the sample came back after sequencing, we found out it was close to 50 kilo base pairs, 150,000 base pairs, and these were close to like from 50 to 70 kilo base pairs. So the size of the head also kind of gives us an idea about the size of the genome. So this is part 1 of the phage project. Then we start with the part 2 of the phage project, which is genome annotation. And what genome annotation basically means is the FASTA file that we receive after sequencing has now to be looked at, and gene names and their locations have to be known. And for this, we use several softwares to come up with knowing what those genes are used for. And again, this is just prediction by computers. So even though we may say that, let's say gene 1 and 2 code for the capsid, unless you do wet lab experiments, we cannot say for sure it is the capsid gene. So wet lab experiments generally have to be done to give evidence that those are indeed the protein, those are indeed the functions of those genes. So basically what happens is, so the first semester when students are doing the discovery process, that's usually the fall semester. And so we send out the genomes to be sequenced. They come back in January or so. And by that time, students are ready for the second semester. And so this is just one section of so many sections that are available, that are there, who do this, because everybody may not be interested in doing this project. It's a totally different biology lab as compared to other biology labs. So a little bit about how the phages are classified. So yes, they are classified into by classical means into Myoviridae and Cephaloviridea. But once the sequence is obtained, phages are now classified in what are known as clusters. And these clusters go by alphabet names like A, B, C, D, and up to, then they can also be A, B, and so on. And this cluster classification is done based on sequence similarity. So once the genome has been sequenced and looking at its size and percentage, they're typically placed into different clusters. Phages that do not have any obvious sequence similarity to what are in the database are typically named singletons. So this is a slide that I took from the Phages DB website, and this is how we carry out the genome annotation. So the first thing is, and I'll start here, I won't go into too many details as to each and every process, but some of the main things that are important, that are critical for the success of this project. First is, of course, retrieving our sequence from the Phages DB site, which is a Phages site. So everything is available there. And then we use this program called DNA Master, which is a software which is free for us to use, for everybody to use, in fact. And the sequence is imported into DNA Master. So from there on, everything that we do is now stored onto DNA Master. The files are saved within DNA Master. So after the FASTA file has been imported, the program carries out what is known as auto annotation. Basically, it just takes the whole gene and annotates it according to the start signals and the stop signals, and basically designates genes based on the those parameters. So now our job is to look at all these auto-annotated genes and figure out whether the program has called them correctly or not. And for this, we do many things. And again, as I said, I don't want to explain all the things that are in the boxes, but just to say that we look at each gene and look at various parameters and try to annotate every single gene. So if there are 150 genes, all the 150 genes are to be annotated. And we get final file that's everything. It's from now on, as I said, once the file is imported into DNA Master, is present within DNA Master. And file has all the notations for every single gene. So once the general file, which has final annotation, is ready, then it is submitted to the reviewers, and this may be then reviewed by external reviewers and then submitted to GenBank. So we do have close to we'll say about 15 to 17 maybe genome sequences that have been submitted to GenBank from our institution alone. So, this next slide is another slide of the annotation process. So So some of the things that we do while annotating a gene is basically, first, of course, is knowing what the function is. So we'd like to predict the functions of all genes, but in reality, we are not able to do that. We can only predict about 20 to 25% of all the genes. All the other genes are, we are not able to ascribe any function to it. So we just say that those are NKF or no known function. So the other things that are done in the annotation process is whether a gene's start codon needs to be changed. And so to do that, we look at the upstream sequences of that gene. Or we need to know if a gene has to be deleted. If there is a big overlap between one gene and the other, that gene is possibly going to be deleted. So those are some things that we do, and this is all done within the DNA master. So you can delete a gene, you can add a gene also. So if, let's say, there's a big gap between two genes, and there's a possibility that there's a start and a stop codon, and that sequence codes for something, so we can add a gene as well. Also, there are some tRNA genes, and those tRNA genes are predicted by using some other software, like the tRNA scan and such. So most of the changes within DNA master are either adding a gene, deleting a gene, changing the start site of the gene, or as I said, knowing the function of those genes. So, the top half are the big changes, and the bottom here talks about if we find a function, what do we, and so on. So again, not talking too much about that process. This is another slide that I took from the Phages DB site. And the first two are the first two columns are talking about the function. How do we ascribe a function to a gene? So to know the function of a gene, we use the program called BLASTP, which I'm sure you all are familiar with. And the BLASTP is used to know the protein function. In addition, we also use this program called spread, which gives us the functional assignment of that gene. So if the E values and the percentage identity is good, then we can say for sure that that protein codes for, let's say, tail chaperone protein or a minor tail protein. So that's so BLAST, HHPRED, Phages DB his own BLAST program. Those are programs that are used to know the functions. And on the right-hand side, which is very important, is this purple column which says synteny. So synteny is basically the arrangement of genes across the different phage genomes, and we find that the genes are arranged in a specific order, regardless of what cluster it belongs to. So, so that's important. So let's say in a phage genome, you find that that order is not adhered to. That means that we need to go back and look at the genes that we have called and see if we made an error or indeed so. So synteny is very critical to genome annotation because it will give us a better idea of calls that we have made. So just to briefly talk about the genome annotation. So the main program that we use is DNA Master, and other databases that are used for supporting evidence include BLASTP, Phages DB's own BLASTP, the HHPRED, the tRNA scan, etc. And so when we are annotating the genome, so let's say you have 150 genes that have been called by the program DNA Master. For every gene, these are the things that we look at, and the notations for each single gene is made within DNA Master. So so every gene, we need to know the start and stop coordinates. Then whether the gene that has been called by the program, is it the longest ORF? Because the longest ORF also means that there's a higher coding potential. So in addition to the longest ORF, we look at the coding potential as such. So the coding potential is known by using another program called GeneMark. And looking at the GeneMark output, we can figure out whether that gene has a coding potential or not. We also try to find out that whether the start site that we have called is being called in other genomes as such. So the program called Starter-Rater helps in identifying the correct start site. Then we look at what is known as SCS, which is the start choice source. So there are two programs that are built within DNA Master, Glimmer and GeneMark. And so we look at whether the program, both these call the same start choice, or if they are different or we need to change it. Then we also look at what is known as D-score or the RBS, ribosome binding site. So by looking at values within the gene, we can figure out whether indeed that gene has a good ribosome binding site, because as you know, that's critical for protein synthesis. Then we look at N, which is nucleotide homology with other genomes, and see and make notations that this gene is has sequence homology with this gene from this phase. And we look at what is the gap. This is the distance between the two genes. The function, which I talked about, that want to know what the function is. So typically, the function genes, or there may be some enzyme genes that code for enzymes or something else. And we also we write what is the function source. That is, where did we know that this particular gene codes for measure proteins? So what is our source, whether it is Phages DB, it is BLAST-P, whether it is HH-PRED or all three. And then synteny, as I mentioned earlier, synteny is also key. We can then say for some kind of assurance that this gene is behaving like other genes in other phage genomes. So this again, it's kind of difficult to name all the different genes that are present within a genome, but some genes that are found in say lytic genes, in lytic phages, are things like lysine A and B. So lysine codes for proteins that help in lysing the bacterial cell. Then this is a holin, creates the holes for the cells to lyse. Then we have major and minor tail proteins. There are tape measure proteins. There are integrase that helps in integrating repressor proteins. So there are at least like a list of 40 to 50 proteins that are now known that are found in different phages. And it depends on the phage you have isolated, which of these genes are present there. So, so in our lab, the phages that we have isolated belong to different clusters, and one of them is the A cluster. So this happens to be the largest cluster, and it mostly has temperate phages. So they belong to the Syphoviridae, which means it has a long tail. And typically, the arrangement of genes on these A cluster genes they're present on the left side, and they are all transcribed from left to right. Okay, so about what we have done at the university is that from the project started in 2013 until now, 1 year or maybe 1 year we couldn't offer this course, and maybe even this year we won't be able to offer this course. And students have discovered 38 phages. So they're all novel phages, and we have them. Of the 38, 16 have been sequenced and femmerated. Femmerated is, so there's a program called Femmerator, which helps in identifying the genes according to their families. So that's so instead of PHAMILY. So this is a gene family as in PHAMILY. And so of them, 15 have been submitted to GenBank. And we have also published 3, and we are on the writing another 1 or maybe 2 from these our discoveries. So these are the different class members that students have created. And so you might see that the phage names are very different than what you would expect. Like if you were to be, learning about different phages. So the students are allowed to name their own. So for example, I remember this name Back Home. Back Home was named so because one of students thought about the virus is the home of the virus is the bacteria. So from that, it became back home. And this one, I know this Midas 2 was isolated by my students. And since it was the first phage to be isolated, and so she kind of related to turning gold. So that was Midas 2. Then most recently, this last year in 2019, one of our students isolated this phage called Shityali. And this is an Aztec word. I don't remember exactly what it meant, but you can see all of these phages have some meaning to them for the students. So what it helps these undergraduate research students is gives them an ownership of the project. So not only do they do some quality undergraduate research, but they also end up naming their phages, and these phages are sometimes, submitted to GenBank. So they also have like a minor publication in their names at a very early point in their educational career. So there's a website called phagesdb.org, and there are currently close to, I'll say 190 or so schools that are part of this C-Phages project. And they are, everybody submits their phages, the lysate to the HHMI C-Phages project, and they are all, their sequence, etc., is all present on the Phages DB site, so we can, access those sites, access those sequences as and when we need them. So, so this is the website, the Phages DB site. I'm going to try to see if I can share, or I probably should not try it. What if the connection goes, something goes wrong with the connection? So this is our website, not our website. These are all the phages that the University of Houston students have isolated, and they are in this phagesdb.org. And so if you click on any of those when it's a live web page, it takes you to the phage page. And within the phage page, you will find out what cluster it belongs to, what's the genome size, how many genes are there, the electron micrographs of the same, and whether there's a GenBank submission. All of that is available by clicking on each of this on a live web page. As I said earlier, we have had 38 phages isolated, of which 16 have been sequenced, 16 have been ephemeраted, and 15 are in GenBank. And we are working on a couple of more that are to be submitted in GenBank. And we are also writing a paper for submission. So as luck would have it, day before yesterday when I was working on this project, I wanted to show you all the arrangement of genes on the chromosome. And it just turned out that this program, ephemerata.org, that the website was down. So I had to literally take a screenshot from a previous a student's poster and use that to show you. Unfortunately, it doesn't have the best, you can't see anything, but I just want to point out some major things that you're able to see. I'm not asking you to read the fine print. So these are two phages. One is called Ku, the other is Midas, and they both are cluster A phages. And so this mauve all around tells us whenever there's a mauve between two phages, this color, it tells us that there is very strong homology between the two phages. Okay, so you can see this is almost identical. And actually, we found out when we received the sequence that it was like only a 1 base pair difference between the two. So, the thought was that was it that one of this the Midas 2 was isolated later, Coog was isolated earlier. So we were thinking about it that maybe somehow it had got cross-contaminated, but it was kind of likely because Coog had been identified the year before. And so it's not possible that we carried some of that and it stayed good for 1 year to cross-contaminate this Midas 2 sample. So it just turns out that it was very close, it was very similar. The other thing I wanted to point out was that if you look at this line, on the top, there are these small colored boxes. These are all different proteins that are FAMs that are called by the program Femerator. Everything that is on the top of this line, of the, that line with the black lines. So those are all genes that are transcribed in the forward direction. And all the genes all the boxes that are below that black and white line are being transcribed in the reverse direction. And this is another website that we use called Pecan. And this is somewhat similar to, since the Femerator, as I said, it had a problem logging in yesterday. So I used this other website called Pecan just to show you that these boxes are basically these are different proteins. And if you look at these two, these belong to the same cluster, EK cluster. You see how closely the alignment of those genes match. So you can see they're also they're not exactly identical, but they're in similar positions, and their functions are also similar. So, so these are the references that I have used. There are a few more that I have not listed. So I'm going to, if anybody's interested, I can provide those references. Finally, I would like to first of all acknowledge all the organizers of this conference for asking me to give this talk. And then this work would not have been completed without the participation of my undergraduate research students. So this is, I keep emphasizing that because when I did research, it was only at the graduate level. I never, I think, did anything remotely close to research in my undergrad years. So we have to give it to these students who come at different hours to take care of their plates, to do their isolations when they are not successful, keep repeating them. So this work would not have been complete without their cooperation. This work was carried out at the Department of Natural Science, where I'm a lecturer, and Howard Hughes Medical Institute and Graham Hatfull Lab. So Graham Hatfull is the PI for this project, and without their contributions, this could not have been done. And here's my contact information. If you have any questions, I'll be very happy to answer them. Thank you so much.
Thank you, ma'am. Thank you very much for an informative session on phages, especially mycobacteriophages and on mycobacteriophages. We have a few questions here, ma'am. So
Sure.
I will read them out to you, and then you can answer them. The first question is, can the technique of probiotics tackle the antigens of the virus?
I'm not sure of that answer, because I thought probiotics are mostly for bacteria. So I'm not sure if they can tackle the antigens of those viruses. I don't think I can answer that question to the best from what I know.
Okay, ma'am, that's okay, ma'am. We'll move on to the next question. Can phage therapy be an approach for mycobacterial infections in human? And if so, how effective would it be?
Yes. So in the beginning, I showed you that this phage therapy has been around from the 1920s, okay, and it kind of lost its value, if I may use the word, when antibiotics were discovered. So from the 1940s onwards, there was a big impetus on using of antibiotics to treat bacterial infections. So at that time, the interest in phage therapy kind of died down. But again, in the last, as you all know, in the last 10 to 15 years, there has been an increase in the drug-resistant bacterial infections such as MRSA, Pseudomonas, and so on. And therefore, these this can be used to treat those kind of infections. So phage therapy has been around. And lately, in at least the couple of cases I've heard, they are one was this girl Isabel's, and there was another lady who talked to us about how her husband had got a very infection on a cruise and how she got phages to basically treat this person. Her husband was on his deathbed until phage therapy came to the rescue, literally.
Okay, ma'am. Thank you, ma'am. The next question is, does increasing the size of the genome have any protective functions or any other role for the phage?
That's kind of a hard question to ask. I think it has to do the evolutionary origin of phages. So, the phages basically are infecting bacterial cells and picking up genomes or picking up genes from one bacteria, moving on to the next bacteria, picking up genes from that. So, if it has any advantage.
Thank you, ma'am. The next question is, how do we annotate genome of Mycobacterium phage attaching the Cas9 genome present in the Mycobacterium?
So I haven't done that, but I'm just taking a guess. You can annotate it simply using software such as DNA Master, because it'll be and then you do a test of the whole genome and find out where the particular gene is, and then you can annotate it from there on.
Okay, ma'am. The next question is, how does the phage identify its target in the host?
So the phage basically, finds a receptor protein on the host cell, and it binds to that. And you probably know from what's going on with the COVID that there are some receptors present on this, like in case, and they basically bind to some. The proteins present on the surface of the phage that binds to receptors, and that from there on it makes the entry inside the host cell.
Thank you, ma'am. The next question is, what are the precautions that need to be taken before the preparation of host cells, that is the mycophages?
So, these are what we call as so we've moved on from using Mycobacterium smegmatis to also Microbacterium foliarum. So the program is now looking at all actinobacteriophages, which includes all the phages that infect actinobacteria. And these are all BSL-1, biosafety level 1 organisms. So we take regular precautions, like as you would working with any bacteria. That is, wear gloves, wear a mask, wear a lab coat, all the bacterial, plates, etc., that have been used, they go into biohazard, and, phage. We just treat it like any other BSL making sure that students are wearing gloves, and, mask is not necessary, but if they want to, we don't have a problem with that. And of course, wearing a lab coat, wearing closed-toed shoes, simple things like having hair tied, and So those are some standard precautions that we take when in any lab, and that's the same that applies to this lab.
The next question is there any possibility of using this phage to treat viruses or viral diseases?
I'm sure there is, like, I don't know, like how they can be treated, but there may be a possibility that one phage used to. I am an expert in that area. I haven't come across any literature about this.
Thank you, ma'am. And now the last question is, what are do you know any free online softwares available to annotate the genome of various pages?
Yeah, so if you go to this website, I can type in, I guess, over. If you go to this .org, and there's a there's a tab there which says resources of something like that, information and resources. And if you click on that, you'll see that there's a software, and the software you're going to be looking at is DNA Master, free for download from the person who go to that. It basically takes you to that website. So DNA Master is a free website that you can use, or a program that you can use.
Thank you, ma'am. With that, we conclude the Q&A session. Thank you very much for taking your time to answer all of the participants' questions.
Thank you so much for inviting me. It was a pleasure interacting with you from here. I hope you have a great evening.
Thank you, ma'am. Now I request Dr. LFE Anand Raj, the Associate Professor as well as the organizer for this event, to give the thanking session.
Good evening. As an organizer of this webinar, it is my duty to thank our distinguished speaker, Dr. Sanghamithra Saha, for sharing her precious time for this webinar. I also appreciate her for making this seminar interesting and meaningful through her excellent presentation. Ma'am, your knowledge in bacteriophage is really awesome. The valuable thoughts that you have expressed on this occasion will no doubt motivate our participants to explore in this new area. I also want to add a special note that our speaker is very kind in supporting our organizing team, which made this webinar very successful. She never bothered about the timings. Whenever we call her for discussion meeting, she made herself available. She supported us every way to make this occasion very memorable. Thank you so much, ma'am. I thank you once again. Thank you all.
Now, I request our professor and advisor for this event, Dr. Renuka Vishwanathan, to come and give the vote of thanks.
Good evening, all. I deem it an honor and privilege to propose the vote of thanks on this occasion, International Technical Webinar on Advancements in Biotechnology. Let me first of all start by giving glory to the Almighty for making today's occasion a resounding success. I wish to express my profound gratitude to our management, St. Joseph's College of Engineering, St. Joseph Group of Institutions, Chairman, Dr. B. Babu Manoharan, Managing Director, Mrs. Desipriya, Director, Mr. Sasi Shekhar, for giving us the opportunity and encouragement in hosting this webinar. I also wish to convey my thanks to our Principal, Dr. V. Seshagiri Rao, and Dean, Dr. M. Parvatavardhani, for their motivation and support. I wish to express my hearty thanks to our HOD, Dr. G. Sri Kumar, for his constant guidance and day-to-day discussions in conducting this event successfully. I wish to express my deep sense of gratitude. Of course, words are not enough for the speakers of this 3-day webinar for their immediate acceptance to share their research findings, knowledge, and experiences which definitely will inspire young minds to pursue their research interests. I wish to express my appreciations and thanks to the organizers, Dr. LFA Anandaraj, Mrs. G. Kalavati, and Mrs. S. Yuvarani for their tireless efforts in making this event a grand success. I take this opportunity to sincerely thank all the department staff members and the invitees and the participants from different destinations for their cooperation and happy learning. Lastly, and not the least, on behalf of the Department of Biotechnology, I wish to express my thanks to the students for their unstinted support in technically managing and for their cooperation to make this webinar a splendid success. Thank you, one and all.
Thank you, ma'am. Finally, I thank the participants once again for attending all the sessions in this webinar. Once again, we thank you all for attending this session. Stay safe and hope to see you all in future events conducted by our department from St. Joseph's College of Engineering. Thank you.